Conservation Genomics Coordinator there at the Botanic Gardens of Sydney, and today I'm going to talk to you about some practical examples or studies of genomics informing translocations, and hopefully that's relatable or usable to you as well. As Andrew has mentioned, genetic diversity is everything, of course. And just because populations need it to ensure they are able to adapt to any environmental changes. But how feasible is it to produce the genetic data to support this and any conservation work associated? The answer is very much so, especially if your study uses our workflow that my team, including Maurizio Rosetto and others, such as some threatened species officers, have helped produce this workflow where it addresses a number of critical questions about a species that would then guide their downstream management outcomes. And so in Australia, there are a large number of flora where data about them is really limited. And so by using genomics, we can gather that knowledge and hopefully use it to help with conservation. And so using our workflow to produce a genetic study is highly feasible, as it would then mean that the results to help with conservation would come in 3 to 5 months' time, which is sort of a good timeline, I would think, to help with the officers. And those solutions that we can provide to officers can include by providing options for creating an ex situ collection with maximal diversity where possible, and also to optimize and manage any existing collections. And as Andrew mentioned, good sort of record keeping is really important. And so we can help screen an existing collection and help find out if your labeling system is on track. As well, we help with any reintroduction work where we find out if certain individuals should be planted together or not, so that to maximize gene flow between genetically distinct individuals. And lastly, reinforcing wild populations so that they have as much diversity where possible. And so currently we have developed or completed, I should say, up to 30 studies on various threatened species. And this is work that's funded by the New South Wales government as part of the Saving Our Species program. This is a large-scale threatened species management program where we want to secure each threatened species in the wild by managing the known threats that those species are facing. And so I will now go through some case studies, including the one that Justin Malley, who's the threatened species officer in charge, is working on. And this is part of the assisted migration project that he got funding from the New South Wales Environmental Trust grant to work on. And this Diploglotis campbellii is the name of the species, and it and others are to be used in this assisted migration project to produce a new population that's on a mountaintop, and that would mean that it's not within the current distribution range, but hopefully would help secure its survival in response to climate change. So how does genomics help, you would ask? So there are a lot of species that Justin has to work on. So genomics would be really helpful to help him prioritize what exactly is needed to be collected in the wild in order to plant in this new population. And obviously not all of the populations would be required if you just have a look at the genomic data. So a bit of background about this species. It has an extremely small population, and all the population is found on private land, which means it's unprotected. And so, and there's very limited recruitment as well. By doing the genomic work on a representative number of individuals of Diploglottis campbellii across its whole distribution, we found out that there's very low levels of genetic variation remaining in the wild, with high levels of inbreeding detected at each little pocket of where the plant occurs. And this is consistent with how unfit the species is presently is. So, and in order to help we address, also this means that assisted migration action is really justified, and to help Justin to prioritize, we then did an optimization analysis on the genomic data to sort of account for what's the minimum population size needed to produce a population that's genetically diverse. So this is what the output from the optimization analysis is showing, with x-axis showing the optimized population size from 10 to 50 individuals, and the proportion of diversity of alleles captured on the y-axis. And 14 individuals seems to be the sweet spot to be selected, and of the 14 individuals, they came from a number of different sites, and it's showing that only 1 or 2 individuals should be selected from each of those sites. And Justin is pretty happy, hopefully, with this outcome. And so this study is demonstrating that we can really help prioritize the on-ground actions as well. This result can be scaled up so that if more funding becomes available, we can expand this translocation population. One thing that, of course, should be bear in mind is to figure out how one should plant these individuals so that we can maximize mating between genetically distinct individuals and minimize inbreeding. So that's one case study. And then now I will talk about Prostantra densa, which is another species as part of the Saving Our Species program. So this species has a really disjunct distribution, as you can see on the map there. And so it occurs near Sydney, where there's urbanized areas present, as well as far north in Central Coast and down south in Jervis Bay. And the main reason why Genomics was requested was to figure out whether it was distinct from another species that looks very similar to it, and that's Prostantra marifolia, another highly rare species. And that's what the photos show, with marifolia on the left and densa on the right. And Genomics was conducted on samples of Prostantra densa collected across its whole distribution, as well as Prostantra marifolia, and we also included some outgroup species to look at how distinct everything is compared to each other. So the split tree results on the left here shows that Prostantra marifolia, which is a cluster in green here, is highly distinct from Prostantra densa, which is the blue clusters on the right. As well, within Prostantra densa, there are differentiated groups that were observed that corresponds with geography. So the genetic groups that were observed were ones that were in the far north, and the ones in the far south. As well, I would like to point out that Helensburgh will turn out to be an admixed population that had that likely resulted from previous gene flow between species, so it was excluded from any subsequent analysis that we did. And so, we also included another test just because this Prostantra densa species was found to be not producing any seeds in the wild. So one question that the officer would like to find out is what is the genetic health of the species? And we did that and detected that there's still some diversity within each of those genetically distinct groups. But in order to help the officer make targeted decision making, we then conducted a series of optimized analyses where we put forth some scenarios that could help with genetic rescue efforts. And so we ran a similar analysis as we did with Diploglottis campbellii using optimization analysis. But this time we are not sure whether those genetically distinct groups would mix with each other and produce progeny that's viable. And so we put forth two scenarios, with one being that we optimize a set of individuals representing each genetic group. So that's scenario number one. And also scenario number two, where we make sure that that population would represent the species overall diversity. In this scenario, it would be good if someone could test, first of all, whether these two genetically distinct groups could mix. But if not, the sort of options we provide can also be used to help with this planting trial. And by using scenario 2, we can ensure that the species diversity is captured, and which is shown here on the graph on the right. Whereas if we move towards just optimizing each distinct group separately, we would capture less diversity. Yeah. So from this study, in general, we can show that genomic data can be used to help sort of with adaptive management type scenarios, where if your translocation is done step by step, it can be really helpful to help target what needs to be done. So I would just like to conclude that genomics is really powerful to help support threatened species management, especially if there are time